Okay, so good afternoon, everybody. Thanks a lot, Clarissy, for inviting me, and I'm very happy to be here and to share with you our experiences with the studies on ancient hybridization in family Zingiberaceae. So first of all, I would like to justify why we selected Zingiberaceae as our model group. Gingers, they include around 1,800 species in roughly 60 genera. All of them are super nice and cute. Many of them are very tasty, and a lot of them are actually economically very important, especially in Asia. They are perennial rhizomatous herbs that are dispersed over the tropics of the world. Some of them are evergreen, and several or many of them are actually going dormant through the dry period, especially when they are distributed in the seasonal areas of the tropics. Our group is working with the gingers over the past 20 years, and it all started with the passion of my colleague, Leong-Škorničková, who started working on taxonomy and nomenclature, especially on the genus Curcuma. And then slowly we started to do molecular phylogenies, study hybridization and polyploidization, also variation in genome size and various correlatives of that specific trait. And after many years, we are still very enthusiastic about this group because we believe that it's a very good model, not only for studies of hybridization, but also, for example, for draft adaptation, as we recently showed in a paper published in a plant journal. But molecular studies on Zingiberaceae were initiated even earlier by the group of Professor John Kress. In 2002, he published the phylogeny based on ITS and MET-K. And this study actually initiated a lot of further studies on gingers. And this phylogeny actually remains a lot of unresolved relationships within the family, as indicated by these red arrows. And we could, for example, blame the markers that they didn't bear enough phylogenetic signal. But already at that time, we saw that there is a lot of cytonuclear discordances, and we could guess that the hybridization may play a role in this family. And today, I would like to show you two case studies, one on the genus Curcuma from subfamily Zingiberoideae, and one from genus Amomum from the subfamily Alpinoideae. And I would like to show you what hybridization, what role the hybridization could play in these groups. Before we start, I need to summarize a little what we know about the role of hybridization, and Clarissy in the morning give a really great talk and the framework. So I will only repeat a little that you know that hybridization is widespread phenomenon in the evolution of plants and animals. But so far, the focus on the studies was on population level or species level, addressing the recent hybridization or current hybridization. Only in past few years, the studies started popping up, documenting also the older cases of hybridization that happened between the ancestors of the lineage which are currently diversified many years ago. Some other studies already documented potential consequences of the hybridization. So this is one nice example from the world of animals, from the cichlid species, where the authors documented that the ancient hybridization can actually trigger the rapid radiation or adaptive radiation. But on the other side of the spectra, there are studies documenting that the result or the consequence of the hybridization can be also like that end of the evolution or silencing of the diversification. From the practical point of view, I believe that we should take care or take into account the possibilities that our species hybridized in the past, especially if the people are using species tree as a prior for their further studies like biogeographies or studies or on adaptation, because it was very nice example from the genus Mimulus, where the authors showed that after detecting the hybridization, the textbook examples were completely twisted over. So my questions for today, and it's actually our long-term questions, is how frequent are or were the hybridization events within the evolution of Zingiberaceae? What could be the circumstances and what are the consequences of this past hybridization, and how the detection of ancient events can impact our current taxonomy? To be able to answer these questions, we need to first detect these hybridization events, which is sometimes challenging because the traces of this hybridization are not that strong as the traces of the recent hybridization. So generally we need a lot of data. In ideal world, we would need probably the whole genome sequencing data or transcriptomic data, but we can also happily live maybe with the HypSeq data at some point, where we can reconstruct the many gene trees, and then via model-based approaches, we can reconstruct actually species networks. Currently, most commonly used methods are modeling species relationships, which are taking models that are modeling species relationships, which are taking into account incomplete lineage sorting, as well as the reticulations. And these models are implemented in tools like PhyloNet or SNAC. The problems with these approaches is that they are computationally very heavy, and there is maximum of 30 species, or roughly what you can squeeze into the analysis to get some results. So now we can dive into the case study number 1. So it's from the genus Curcuma, which includes around 140 species. The species are usually dormant, economically important, but what is important for us is that the genus itself underwent very complex evolution, because the hybridization and polyploidization is very common. Currently, there are very unclear, we see the unclear species boundaries. There is many different species concepts, and we are still resolving many taxonomical and nomenclatural problems. We do it or to do so, we are actually using around 1,000 targeted single copy genes that were specifically selected for the gingers and for curcumas. And with them, we are trying to fine-tune the phylogenies, checking how many hybridization we have within the particular lineages, also checking how frequent are the hybridization between lineages. But today, I would like to put your focus on what we resolved on the deeper level of the evolution, while analyzing these HypSeq data with the PhyloNet. So we observed at least 3 ancient hybridization events. 2 of them led to the lineage diversification, while 1 of them resulted in kind of like silencing of diversification. In more details, the first or the oldest ancient hybridization happened roughly 14 million ago, where the ancestor of genus Pyrgophyllum introgressed the ancestor of the genus Curcuma. Both ancestors probably have the similar or same chromosome count, and after this hybridization, the lineage of or the whole genus Curcuma basically diversified. This event actually nicely explaining previously low or no supported relationship of the curcuma and its outgroup species, as published by Kress and also by us later on. Very similar case happened several million years after, where the ancestor of the Rosciana lineage introgressed the ancestor of the Curcuma lineage. Again, the ancestors probably had, or most probably have had the same chromosome number. And again, after the hybridization, the lineage, the species lineage of the genus Curcuma diversified. And we believe, or we hypothesize, that actually this hybridization event was followed by the spread of this lineage to Indian subcontinent, and also it was accompanied by niche diversification. About this topic, my colleague Marcos, he will give a talk a few minutes later. The last case from the Curcuma is different because the introgression went from the ancestor of Sabenicheniopsis, which has completely different chromosome count than any other Curcuma lineages. It's very messy and nobody knows what's happening there. And this ancestor introgressed the ancestor of Rosciana lineage. So the chromosome counts were incompatible, and the result was some lineage which has currently only one extant species, Curcuma vamana, which is sexual but behaves very weirdly, and it's still super enigmatic what is this species. Also biogeographically, it's very hard to understand what happened, but the take-home message is that it's the break of the evolution. This is the case study 2 from the genus Amomum, which is a little different story. The genus is smaller, it has around 60 species. The species are evergreen, and they are distributed all over the Indo-Malaysian region in the seasonal areas as well as evergreen areas. And what is interesting about this genus is it's having the great range of genome sizes, starting from 1.7 thousand to 7.6 thousand megabase pairs. And our primary interest in this genus was that we wanted to study the adaptive role of this genome size. Very briefly, the genomes of the species are increasing from the bottom parts of the phylogeny to the terminal ones. And we hypothesize that it also has something in common with this distribution, because the species with the smaller genomes occupying the seasonal areas, while the species from the terminal clades with the biggest genomes, they are everywhere. Interestingly, this clade of the species with the largest genomes are of ancient hybrid origin. And again, the ancestors of that clade had probably the similar chromosome counts, and we believe, hypothesize, that there was some coincidence of that hybridization and genome size enlargement. This is very new results, which probably will be published soon. And we actually analyzed the repeatome of the several individuals from the genus Amomum. And we found some correspondence of that hybridization event and proliferation of some particular repetitive elements. And now we are really keen and interested if we could find some causality between the hybridization, repeat element proliferation, genome size increase, and ecological characteristics of this group. Now I'm going back to the whole family level. I probably need to say that we are actually finalizing the new phylogeny of the whole family based on these target enrichment data. But funnily, the backbone of the phylogeny is still poorly resolved, and it's still poorly resolved in the same for the same lineages as published by Kress et al. So we started to do these phylon networks also on these big trees, but we need to subsampling to get the analysis running. But I can tell you that all our preliminary results suggesting that each of these poorly supported lineages are actually connected with that ancient hybridization events. So to conclude and answer the question that I indicated before, regarding the frequency, we believe that the ancient hybridization is very frequent, at least in our family or in the family Zingiberaceae. Regarding the circumstances of this hybridization, we believe we need to study more the karyotypes and the similarity of the karyotypes, because it seems that the more similar karyotypes, the bigger chance is that the lineage, which is of hybrid origin, can diversify, while if the karyotypes are very different or too different, then it ends up as a dead end of the evolution. Beside this diversification or dead end of the evolution, other consequences might be like the genome size changes, different repeat proliferations, chromosomal changes and rearrangements. From that case of curcuma lineage from the genus Curcuma, also the polyploidization was much more frequent after the hybridization, and probably also the evolution of the new niches. How we deal with this, or how we, what is the impact of detection of these ancient events for the taxonomy? I would here somehow agree with the Clarissy that I would not take these hybridizations as a problem, because I believe that it actually helps us to resolve the problematic relationships. But it might be still problematic to somehow do the taxonomic units, or how to call the taxonomic units that are of hybrid origin. So this is what I cannot answer. So these are my acknowledgement. I would like to thank all the people who are helping us in the field. These are the super nice people from Vietnam, from Thailand, from Edinburgh especially, and also from the US. I would like to thank everybody who helped us in the work and taking care about our living collections, for funding, of course, and you for your attention.
Now we have some time for questions. Okay.
Thank you, Eliska. Very nice talk. It's amazing to see this kind of cases where you have less diversification after hybridization, because this is quite rare in the literature. Congratulations for spotting one. But you refer to it as a dead end. However, this lineage is stable for millions of years or quite a long time, even if it didn't diversify. I don't think it's a dead end, right?
Yeah, I mean I could not find any like other way how to call it. Like it's kind of like not diversifying. Maybe that's better. No, this is the about the chromosomes. It's actually that specific case of that one species that really it's combination of completely different basic chromosome numbers, and it never happened anywhere else. So it's there is one lineage which you cannot really say what is the basic chromosome number. So we don't know what's happening, and we need to search if there is some similarity, and it's really mess. So I believe it's the result of that mess.
Well, thank you very much. Very nice. So we have one positive talk about hybridization now. Let's see how it will be in the end. My question is, do you have any knowledge about the current hybridization that people doing artificial, or you find hybrids in the nature, and how is the reproductive isolation? I would like
Yeah. No, that would be talk for maybe 2 hours, maybe 5 hours. So yeah, that's definitely like very common. So I mean, I showed this particular lineages. So within each lineage, there is many species complex. In one lineage, that is full of polyploids, which are also the results of the common hybridization, and artificial for sure. So the people are using the hybridization to do a lot of like horticulturally and economically like valuable species, and and they are hybridizing even like species from completely distantly related lineages. So it's like between subgenera, and I believe they could do something even from sister species. So it's like the barriers, the reproductive barriers, are probably not there, not too strong. Yes.
Fantastic talk. Thank you so much. And I have so many cool things to think about. But one of the things that kept striking me is that, we know within the Zingiberaceae, especially within the Alpinioideae, that Amomum and Alpinia are such complicated groups taxonomically. And in fact, one time I asked somebody how they knew whether something was an Alpinia or an Amomum, and they said, well, it's because I know the species name. Which indicates that there's no morphological characters. So do you think that there's a relationship between this ability to have sort of deep level hybridization events and the lack of really strong morphological characters that we've been able to use to define some of these clades?
Yeah, I mean, I would say like firstly, yes, but of course we need to, because there are these like indicators of ancient hybridization also for the Alpinoideae. Also, it's not that many as in Zingiberaceae, but I might guess that in Alpinoideae there could be also more like these rapid radiations or something like this, because it's probably the combination. So yeah, I would, yeah, that would be my answer.
Any other questions? Okay, thank you very much.